Thank you so much, Namshik, for your short talk about repurposing drugs for COVID, a very topical issue that a lot of our leaders of tomorrow are really interested in. My first question is, could you speak a little bit more broadly about the overall vision for the Milner Therapeutics Institute and the work that you're doing there?
Sure. Oh, thank you. Yeah, so as you know, the Milner Institute is an academic institute under the Cambridge University, and we are doing some kind of research for the therapeutics or drug discovery. But we have some slightly different concept than the other research institute under the university, which is we are really looking forward to the open innovation and also doing a lot of translational research. So we do have a consortium in the institute called the Milner Therapeutics Consortium, which is more than 80 organizations under the umbrella. So there are some academic institutions, but also our partners in the industry. For example, GSK, AstraZeneca, Pfizer, and Johnson & Johnson, like large pharma companies. But also we do have quite many of SMEs in biotech area. Some do AI data science like my group, but some do developing a antibody, some do the cell therapy. So there are so many different type of companies under the same umbrella. So we are really working together, academia to industry, industry to academia, and we really working toward to making a new or better therapeutic targets. So yeah, that's what we do.
Fantastic, great, really novel innovation model. We had a question come through the Slido. Given the similarities between the genome of SARS-CoV and SARS-CoV-2, have you applied the study to SARS-CoV?
Yeah, that's very interesting question. Yes, everybody knows there is a lot of sequential similarity and structural similarity, but two different virus having quite different symptoms. So yeah, that's a very good question. So we actually are now trying to do this kind of comparison between two, but we haven't finished the full analysis, but that's ongoing analysis in the lab.
Great. And another question is there a cell-specific context to this gene expression profile and drug simulation, since defining cell specificity is needed to prevent undesired effects?
Sure. Yeah, that's another very good question and important as well. So maybe everybody knows that this kind of a lot of effort, really incredible effort, to be honest, in the history since the lockdown or the pandemic happened. So many scientists work really hard, but I think now it's fine. But when we just face this kind of new disease, everybody kind of in real hurry to make some kind of breakthrough. So at that time, a lot of groups are generating the data, but that's coming out from some different cells because they just want to do it quickly. So some data coming out from colon, some data coming out from lung, but I don't think this is not really bad, but I think this is not completely ideal. So if we really wanted to do more cell-specific ones, then we need to carefully design the experiment. So what I meant is like we couldn't get this kind of fully controlled or fully managed kind of data per each cell type, but now it's available in the literature. So what I meant is like we are going to do it, but also there are many groups also in Cambridge or in somewhere in the UK. They are already applying for funding for this kind of cell type specific experiment. So I hope we will have better and more data later this year, and then we can really fully working on cell specific ones. But at the moment, we can't do it because of the data availability.
Mm-hmm, mm-hmm. And I think a little bit related to that, but what are the challenges that exist when integrating both proteomic and transgenic data sets, and how can we address these problems?
Yes, I think this is really a good question again. This is actually one of the things my lab mostly doing, like so-called multi-omics integration. So as you know, multiomics is not only from proteomics and transcriptomics, but there are like metabolomics and also epigenomics and genomics. So this is a kind of million-dollar question. Like in the end of the day, we want to understand how the genes are expressed and how they are regulated, and then how they can make a protein in a right time and right place. How that happens. So entire mechanism is the kind of question. So now, fortunately, there are a lot of revolution in the experimental side, and a lot of different data is available. That's kind of happy problem. But also, as you asked, this is kind of really important question: how we can properly kind of integrate or properly analyze these kind of quite different data sets for just one question. So actually, in the lab, we are using quite many of mathematical modeling, or sometimes using machine learning to properly kind of pull together all the information from the beginning. What I meant is, like, of course, there is a number of ways, like you can analyze proteomics and transcriptomics and some other data type of multiomics, and then combine the result at the end to understand the mechanism. But what we're trying to do is, like, we kind of making mathematical model to put each kind of component in the mathematical model from the multiomics data. And then we wanted to more systematically analyze this data set. That's kind of very interesting question. And also, that's one main area where we are working on.
Excellent. So do you have any closing thoughts or ideas that you'd like to share with our young leaders of tomorrow from around the world before we move to the next short talk?
Sure. I think well, as a computational biologist, we always think about the data, but these days is not only us, which is great for us, but also there are many things we need to think about, like what kind of data, what kind of quality, how we can make it, and what this kind of data actually tells us. So what I meant is, like, we need to think about more carefully when we're making the data. But also, in another hand, it's very important to using how we can using the so-called AI or machine learning. I mean, AI or machine learning is not always needed for all the kind of data sets. Sometimes just simple statistical test is enough, or even is even better than just using machine learning. So I think there are a lot of kind of advantages of the machine learning, but sometimes we need really thinking about what kind of data is in our hands, and hence what kind of analytical tools are needed. So this is kind of very important question too. So yeah, that's kind of piece of advice that I can give.
Great, thank you so much. The right tool at the right time, I guess.
Yep.
Thank you so much for your time, Namshik.